students. Today we are going to continue the chromatography techniques. Today we are going to discuss about the GCMS and the LCMS. So in here GCMS, in the sense it is the high and powerful analytical techniques. It is one of the physical one. So in here there are two techniques, they are combined to form a single method in order to analyze the mixture of the chemicals. The GCMS, it is an instrument technique comprising a gas chromatographic coupled, and it is also in a mass spectroscopy also. So they both are in order to analyzing, both are combined in order to analyze the complex of the mixtures like chemicals. It may be separated, identified and quantified. In order to compound to analysis by the GCMS, it must be sufficient, volatile and thermally stable. Because we are here in we are produced in a high temperature way. So this is the instrument. So this is in the parts of the GCMS parts. What is the history of the gas chromatography and the GCMS likewise? So in 1952, James and Martin, they separate the mixture of the small carboxylic acid. And in 1958 by Coley, he was introduced the open capillary columns in this. And in 1976, Dantune and Jerner, they both are introduced the fused silica capillary column. They may be considered as a breakthrough the development of the gas chromatography. It is most important process. In 1950, Ronald and Frick, they both are used the mass spectroscopy as a detector in the gas chromatography. The same wise, the technique is mostly used the sample which are mostly stable in the thermal. We already discussed it. So in here they are mostly thermal one. They will be used in. So in GCMS, they are mostly applied in the drug detection, fire investigations and environmental analysis likewise. Especially in 1970, it may be most applied in the samples which are obtained from the planet of Mars during the mission. And it can be also used in the airport to detect the illegal substances in luggages or in human beings. The principle in the sense the GCMS, they mostly work on the principle that the mixture will be first separated into the individual substances when they are heated. So that's only our substances most viable in the thermal one. And then it will be heated by the gases which is carried through the column. They will be inserted along with the inert gases, that is such as the helium gases. At the separated substances emerges from the column opening and they are directly flowed in the mass spectroscopy. So thus the facilitations identified of the individual substances. In the workflow, in the sense, first we have to sample preparation. So in sample preparation is involved the extracting and preparing the sample for analysis, ensuring the proper handling and preservation of analysis before the injection into the GCMS systems. So that only it will be give a good result for us. For an instrument, in the sense, it will be we have to give a proper column temperature and the workflow rate and the ionization model of the optimum separations for the detection of the compounds. And data acquisition, in the sense the GCMS, they are recorded the mass spectroscopy generated by the mass spectrometer for each component issued by the GC columns. Their capture and retention times and the peak intention. So it will be in a safe manner. And the interpretation. So after finally we get the acquisition, we have to interpret the data which will be given by that. So that will be involves analyzes the mass spectrum, identify the compounds based on the fragments pattern and the quantifying the target analysis in the sample using calibrate curves. We already seen the GCMS is mostly applicable in the most of the field. So in here the common one, the forensic and the criminal investigations, environmental analysis, illegal substance detections, chemical agent detections and petrochemical analysis. So not only this, these are the more widely used, the applicable in the GCMS for in our day-to-day life too. The next one, liquid chromatography. Liquid chromatography, in the sense it is a LCMS. So in this one, it is also the combination of the liquid chromatography as well as mass spectroscopy. Likewise, the GCMS. So in the GCMS, in the sense we have used the gas. In here, instead of the gas, we are used in the liquid in here. So here there are separation, identify and quantify the components in the complex mixtures. It provides a high sensitivity, selectivity and versatility in the analyze the wide range of compounds, making a powerful tool in various fields such as pharmaceuticals, forensics and environmental sciences too. So here also the same thing, the separation process. So liquid chromatography, they are separates the components in the mixture based on the differences in the interactions with the stationary as well as the mobile phases. So that only to allow the or isolation the individual of the components. And retention time. The retention time, in the sense the liquid chromatography refers the time taken by the component they are travel through the chromatography system. It provides information on the interactions between the stationary phase. Ionization process. So in this one, the mass spectroscopy, so the ionization converts analyzes molecule that into the charged ion for the separation and analysis one. So in here, various ionization techniques are available based on the type of compound being analyzed. In mass to charge ratio, they are measured the mass to charge ratio ions to enable precise identification and characterization of the samples. The instrumentation, what are the workflow in the sense? So in here the LCMS technology, it involves by using the HPLC, wherein the individual components in the mixture, they are first separated followed by the ionization and separation of the ion based on the mass charge ratios. The separated ions, they are directly to the photo or electron multiple tubes detector, then which will be identified and quantified based on the each ions. So the ion sources in an important component, they are in many MS analysis, that is mass spectroscopy analysis, and this basically aids in efficient generation in ion analysis. So the ionization molecules, it mostly used as the sources are APCI or it may be ESI. They mostly commonly used the one. They are quite like GCMS only, but they both are GCMS as well as the LCMS. They both are the same goal only. In order to substances are identify what are the mixtures, what are the substances they are mixtured in one. They are identified by the separation and mass spectroscopy measurements. So in the LCMS, they are high performance liquid chromatography to separate the substances in the sample. They are much like a gas chromatography, except it use of the liquid mobile phase. So instead of the gas, in here they are used as a liquid. And also the another technique, the GCMS, they are involving the heat one, but in here instead of the heating the mixture, HPLC uses as ionization to separate the ions based on the M/Z, that means the ratio, and then introduce them into the mass spectroscopy for identification and quantification. So these are the instruments and then types. So this is also mostly widely used one in our day-to-day life. Basically biotechnology, environmental monitoring, food processing, quality controls, pharmaceutical quality control, forensic analysis and bodily fluids. Bodily fluids, in the sense they are mostly used in our body production liquids. Okay, up to this. What is the differences between the GCMS and LCMS? We already know that they both are the techniques, they are same one, but there are the differences in here. First one, they are GCMS. In here the gas mobile, it will be used in the GCMS. Instead of the LCMS, they are used as a liquid one. the same, there is a method, and also the while using the liquid or in the thermal one, in here they are used as a ionization method in order to separation one. Okay, so up to this I conclude my work. Thank you.